Molecular U is a platform that we developed to really enable our ability to see disease before symptoms appear. So we can get in front of it, we can predict where an individual is going to go in their health and support them in that journey. And this happens to be a pretty good time for things like diagnostics. There's this whole renaissance going on, and it turns out that we are actually very well suited. We have a proprietary lab-developed test. We are, all of that data supports clinical decision-making. We're leveraging multi-omics, very similar to nobody else. In fact, we have a unique platform. We are AI, but our AI is human-supervised, and we are data-first. And we're working on this strategic entry into the reimbursement market so that we can amplify our returns. So this was an article that was put forward by the Illumina Venture Group, and really to show that this whole diagnostics is coming around again, and there's a lot of value in this market. So we've got a world-class team, truly. I'll allow Jim to introduce himself when he speaks. I'm going to focus on the science, and Jim will give you more on the business side. Gene Skolnikov is our Chief Technical Officer. He's got a rich background in pharma, but also in startup. Murdoch Kalehi is our Chief Medical Officer. I myself am a scientist by training. That's why I get the title of Chief Scientific Officer. I founded Molecular out of a personalized medicine initiative, which I started after a career in pharmaceutical and biotech, where most of my career had been focusing on understanding disease. What are the drivers of disease? How can we fix it? And then it really became clear to me that we really didn't understand the human condition very well. So we started this thing called the Personalized Medicine Initiative with the idea that we could design therapeutics that would be specific for that individual, not just the disease. So in that journey, we identified multiple technologies, and we tested all these technologies, and I'll get into that a little bit more. But the proteome and the metabolome were the ones that gave us the deepest understanding of what was going on in an individual right now, because the proteins are the functional element of every biological system, and the metabolites are basically the reporting out of what's going on in that biological system. So we're fortunate to have two world leaders, David Wishart, who led the Human Metabolome Project, and Christoph Borchers, who was really one of the top analysts in proteomics, who's given us the quantitative measures and allow us to multiplex our tools. So let me just give you an example of the power of this technology. So this is Diane Ballin. Diane has given us permission to share her story. Diane joined Molecular U in 2018, primarily to stay healthy. She was a health-conscious individual, and then she would do an annual assessment to see how she's going over the years. In 2022, she had a number of biomarkers that aligned with biological pathways that pointed to a very active neoplasia, particularly pointing to an association with pancreatic cancer. So we shared this with her practitioner. The practitioner treated it like a vital sign and ordered imaging. The imaging identified these small lesions on her pancreas. Normally they wouldn't biopsy because they were so small, but because of the data we provided, they did the needle biopsy, and it turned out that it was stage 1 pancreatic cancer. She had no symptoms. It was all based on this data. Now, I don't know if anybody here has got anybody that has had pancreatic cancer. It is the worst cancer you can have. It has a very low survival rate, but not because it's not operable, because it's caught too late. So she had surgery. The surgery was successful. We removed the tumors, and you can see that at the year after, the biomarkers basically returned to normal. She did have a damaged pancreas, which is affecting her metabolism, and she lost her spleen because of the vascularization with the pancreas. By year 2024, though, she's fully recovered. So this is really what we're about. If we wait for symptoms, it's typically too late. And so if we can develop that technology that allows us to see disease before it becomes symptomatic, we can get way upstream and we can make changes in lifestyle, which is 70% of our biology. Or we can pick up those cancers and have them removed before they become heroic efforts in order to save. We can also use this technology to support patients that might already have a primary condition, so we can give them a much more personalized care path, so we can target what's most important so things don't progress. Now let's go back to this concept of multi-omics, and it's much longer than this list, of course. And we've actually looked at the microbiome as well, which kind of falls into the genomic side. But Watson and Crick really unraveled all of this for us. They gave us the genetic code, which allowed us to understand genomics, which gave us the primary sequence of our proteins so that we could really do all the other omics that we're doing today. But genomics is fantastic for understanding primary structure, how things might be regulated, but it doesn't help us understand risk except for risk over a lifetime, which is very hard to action. Epigenomics and transcriptomes is really cell-specific, so it's very hard for us to access and measure. But the proteome and metabolome, as I mentioned, those are the real active ingredients in every biological system, and they actually enter into our bloodstream, and we can use the blood as a window to what's going on in the rest of the body, and that's what we've done. Told another way, this individual has the same genetic code, identical, yet the phenotype is very different. It goes from a creepy, crawly caterpillar to this magnificent flying butterfly, not because of its genetics, but because of which proteins are expressed and what those proteins are doing. So our platform is a two-sided solution. We start with a single draw of blood. We only need 250 microliters of plasma, which we then quantitate using a proprietary assay platform in a lab-developed test, so everything is clinical-facing. We've gone through the heavy lift of getting this first multi-omic test in a CLIA COLA environment. We also have ISO 15189 certification, so we can operate pretty much anywhere in the world. We've selected our first 288 metabolites and proteins, high value, that give us that mechanistic insight into how the body's functioning so that we can actually get ahead of disease. And then we take all that information and we put it through our AI data technology. Now, we haven't developed the instrument that you see up here. That's a mass spectrometer. Everything is run on a mass spectrometer. There's huge advantages to the mass spectrometer because you get absolute quantitation, but you can also multiplex. And so right now we're doing 288 metabolites and proteins, but soon we're going to, at the end of this year, it'll be over 1,000. And that'll help us get deeper into the conditions that we're looking at and broader into more cancers, et cetera. The other advantage is that everything, when you're comparing the same sample, so you're not changing samples all over the place, you're not re-sampling from your blood, and you're all on the same scale. So it makes the data interpretation much more straightforward. So first step is we roll this up into the biological pathways. This helps us understand what is really going on at the molecular, cellular, even smaller level. And then from there, we can take all those biological pathways and we can get a much higher level to see what are the risks that an individual may face across all the functions. So this is the most comprehensive analysis you can get from a single draw of blood. On average, we have an 88% predictive value, and I'll give you an example of how we calculate that. Today, as I mentioned, we're at 288. At the end of the year, we'll be over 1,000 biomarkers to get us deeper into the cancers. These are the cancers we can do currently, and these are cancers we've actually detected in our populations at early stages and variable stages, to be true. So let me give you that example. So we calculate our accuracy using ROC curves or radio operator curves, which were actually used originally in the war to determine how sensitive your radar is. You want to be able to sense enough to pick up the birds, but you have to be able to differentiate between the birds and the planes so you don't bomb the birds, right? So this is how every test is determined on its accuracy or predictive value. So you can see that we've got an outstanding predictive value for Alzheimer's disease. And what's more interesting to us is that each of those biological pathways is well documented in the world's literature as being drivers for Alzheimer's disease in combination or not. It really varies, and I'll give you a bit more information on that. So this is using that same platform to look at patients when we entered into a partnership with a dementia clinic in Florida called Gray Matters. They gave us, this was blinded originally, but they gave us 74 patients, 5 of them with Alzheimer's disease, 13 with mild cognitive impairment, and 56 which were considered essentially healthy. We reclassified that patient population by using our single assay on all of these patients. 4 of those patients were absolutely Alzheimer's disease, but 1 of them was definitely not, had none of the hallmarks of Alzheimer's disease, looked much more like vascular dementia, which is a totally different care path. The others we found across the board, 31 of the patients within the Gray Matters actually were at risk, at varying risk for Alzheimer's disease. And then 38 of the healthy actually had a number of conditions that needed to be addressed that weren't necessarily around Alzheimer's disease. what was even more interesting to us is that we could not only say who was at risk, we could say why they were at risk. And so we could basically stratify the population into 3 endotypes, so 3 different source drivers, if you like, of the disease. So one for inflammation and oxidative stress, the other more on the transmitter metabolism, the third more on the dyslipidemia. The huge advantage now is we've designed care paths specific for each of those populations. So we have a much better chance of success. I'll give you one more example of an individual, a 7-year-old female we actually sampled just before Christmas break. And we're probably the only test that can actually pick up depression physically, so using blood biomarkers. So we determined that our highest risk was depression. When we interviewed her and went through her data with her, she told us that she's actually clinically depressed. She's on antidepressants and they're having no benefit whatsoever. Why? Because they're treating the symptoms, not the core of the condition, which is this neuroinflammation. So we're changing her diet, we're helping her get off her junk food, we're getting her off her butt, starting to do some exercise so we can reverse all this. But what was a little bit more concerning to us is that in fact, she was also at risk for what we saw primarily as colorectal cancer. There was a possibility that it could be some other cancer, but most of the biomarkers aligned with the colorectal cancer. And you can see that she had a number of biomarkers that are well out of range. So during the interview, she explained to us that after she had her molecular U, she had her colonoscopy. So we're right on and picking up the fact that she did have some tubular adenomas that were removed, plus some polyps that were removed. So we tested her a second time, and you can see the second time in 2026 that most things have come back into range. However, she's not fully out of the woods. She still has some risk of cancers, so we're going to continue to follow her and make sure that the changes that we're providing her are helping her. But as I mentioned, we do provide these action plans that are mostly around lifestyle, and they do work. We get about a 50% increase in the health scores when people follow their action plans. So lifestyle is important, and we can do a lot if we can pick it up early. So I'm going to hand it over to Jim now, and he's going to share with you how we're scaling the company and this exciting technology.
All right, thank you, Rob. So maybe a little bit about myself. I've done 7 digital health companies since the mid-90s, back when dinosaurs stomped the earth. My biggest success was WebMD. I did the consumer-facing business there, and I was a VC for a while. So this is a really powerful platform, and the biggest question we have to answer sometimes is where to start. So we have a pretty good plan here. First of all, stepping back for a moment, there's a global wave of people deciding they don't want reactive healthcare anymore. They want predictive. They want to be ahead of things. It's generally the top 15% of any population. And this is a snapshot it's provided by Global Wellness. It's a $6.8 trillion economy and has double-digit growth rates. And we play in all these circled bubbles here you can see. We consider ourselves to be foundational infrastructure for prediction, personalized medicine, physical activity, nutrition, all the things that you see there. So how to scale that as a small company, we decided we were a franchise. So we're a multi-omic franchise, and so where we create value is in our platform. So the things that you see on the right are things that our platform provides, and the things that we ask, since all healthcare is intensely local as far as this composition of private, public pay, reimbursement, different demographics, et cetera. We look for really qualified management teams that want to take this forward and also have access to capital. And I had somebody initially ask me, well, why don't we just do it ourselves? And I said, knock yourself out. If you have $30 million and can round up the top 5 scientists in multi-omics in the world, and 3 years, you'll be where we are now, but we'll be 3 years ahead of you. So why don't you just save yourself the fuss, use our platform, and you be good at what you do. And so I haven't had that statement thereafter. But here's what our platform provides. And this also distributes benefit to the rest of the world's network, because we really want to change medicine. We found every single market has little benefits that we can vacuum up and then redistribute globally to all of our partners. So it's a really good model for the smallest things. The top, we manage the assay evolution. As Rob said, we're going to go to 1,000 biomarkers. Version 3 is already being planned. We'll be at 1,500 next year. We do all the QA. This is a super finicky process. So to replicate it, we make sure we have a constantly updated book of SOPs, et cetera, and we manufacture and distribute the assay. On the bottom, we also try to provide more knowledge management around regulatory and compliance, operations. We write all the software. We don't give up our data model. Every time that we discover one of those heat maps that you saw with Diane, we patent that. You can't patent biomarkers, but you can patent algorithmic relationships. So we think that we're on a race right now to find and discover all these key nuggets out there in human biology. We've also sized up the markets. We actually do the top 20. I'm only showing the top 10. We're active right now in Canada, the US, South Korea just launched last month. We have Switzerland with an MOU with a really fantastic local partner. We're flying next to the UK, which is very similar in a lot of ways to Korea. And then we have a really strong group out of the KSA, Kingdom of Saudi Arabia, and we're going to be, they've basically just gave us their desire to go ahead, so we'll be there in May. And then we provide them a playbook of how to commercialize based on our experience. So number one, private pay is always first. People want to pay out of pocket. Reimbursement's always slower, but one thing we've discovered is if we, for example, I'll show you reimbursement in a second, but if we have a study that shows how to predict side effects for cancer, we can do that in one market and then distribute the benefits globally to all our markets. So what's that playbook look like? Well, we have things in fitness and performance. In the modern economy, a lot of people see their trainer more than their doctor, and trainers are actually becoming almost like first-tier healthcare providers. And then you have concierge medicine with longevity, and then kind of double-click into subcategories that you see on the left. And we just kicked off reimbursement in the US, but we're also going to concurrently do this in Korea. First one is precision prior auth. We're really good at autoimmune conditions, for example, rheumatoid arthritis, and getting that right beforehand avoids a $30,000 to $60,000 a year drug. Oncology, there's known side effects from checkpoint inhibitor therapy, which again is autoimmune conditions, so we can piggyback on the first one. We can actually, with a high degree of accuracy, we just did a study with McGill, where we were 80% of the time able to predict side effects that would occur in patients as a result of checkpoint inhibitors. MRD post-treatment, the person most likely to get cancer is somebody who's already had it. And then lastly, you've seen our Alzheimer's data. Lastly, we have partnerships. We have tons of inbound, so we are doing Vancouver Fire Department. Occupational health is great. They get exposed to a lot of toxins and are very worried about their health. Gray Matters is the Alzheimer's clinic. Nationwide is one of the largest children's hospitals in the US, and we're doing a partnership with them in rare disease. The middle are resellers, and lastly, we're doing some clinical trials. So financially, we'll have good, stable growth in private pay, which is the bottom one there, and you always contend with this hockey stick phenomenon, but that generally happens in reimbursable events because you end up getting a lot of money. It's not a ton of tests. You can see it's 1,750 next year with the results of our first reimbursement studies, but it grows pretty quickly. So that's where the big upside is we have a foundation on private pay, but reimbursement really kicks us off. We're doing a $15 million Series B raise, and it's all going to activate our markets globally and within those global markets. We want to support our partners. Just to give you an example, this was what we did in Korea, and we're about to close this offering. So we did a Korean securities offering. We were 30 million pre and raising 10. It's been oversubscribed. We look to close that in the next 2 months. And then I'm going to close. This is kind of almost a PhD thesis, so don't try to read it all. But I will say this is what we're providing to Thermo Fisher, Danaher, Agilent, LabCorp, Quest, all the big instrumentation manufacturers. Is this has nothing to do with us on the left, but we're about to transform mass spec because of what we're doing. It's never been applied to clinical medicine. And we're basically going to bring them in as partnership money and investors alongside of whoever we have leading our Series A or Series B. But last year, that's a good way to potentially get acquired. There are $60 billion of acquisitions in the multi-omics space. That's more than 10 years combined before that. So this whole area is on fire. All those companies I just mentioned have at least $500 billion of capital and dry powder to use to go buy things, and they're super aggressive. They haven't accounted for the wellness economy takeoff, so that's another one. And we think by ourselves, we're going to drive the sales probably of 335 mass spec setups next year or in the next 5 years, and that would be about a $500 or $600 or $1 million run rate for any equipment manufacturer. So the stakes are high for them, and that's why there's interest there. Other than that, happy to take questions. Thank you for your attention.